Hello everyone. It's a great pleasure to be here. I'm actually based in San Diego, California, so I'm near the headquarters of Life Technologies. So normally I cover Southern California, Arizona, New Mexico. So it's really fun to be down here and get away from from my own work right now. I have to say I am a little tired and disappointed in myself because not because worried about this presentation today, but some of my Argentinian coworkers took us out last night for dinner and we went and saw a tango show, and I just have to admit I was up all night dreaming about being a tango superstar. And I quickly figured out, after practicing a couple moves in the room, I'm not going to be a tango superstar. So at the most I can do that. So anyway, we'll all agree that I should stick with my day job. And I'll present to you, one of my babies in the company. I've been associated with a product called the OpenArray. It's a nanoliter qPCR technology. And we have our new system now that's called the QuantStudio 12K Flex. You've heard earlier today, the discussion of QuantStudio and how it actually ties into genetic analysis, how it actually relates to, ion torrent, PGM, next generation sequencing. So typically when we do these talks, I actually come after the next generation sequencing part. And the reason why obviously is because this is a qPCR technology, which is a validation platform. So in the workflow, in the paradigm of genetic analysis, of course we're always starting with hypothesis-free discovery on an ion torrent platform or a next gen sequencing platform. And then you come back afterwards and do a more refined pinpoint validation using qPCR technology. So to start, I always like to start off a little bit with a TaqMan overview of the chemistry. Many of you are probably familiar with TaqMan chemistry and maybe the differences of that assay as opposed to a generic labeling approach. So I'll start off talking about TaqMan, and then a brief introduction into the OpenArray technology in general, and how it flows into this new QuantStudio system. And then I think the bulk of the talk, we'll talk about the machine in general. You saw the instrument out there. It's a very nice looking big box, and we'll describe in detail all the features, the ins and outs of not only the hardware but the software as well. And then quickly talk about some applications, I'll show you some screenshots of posters that I'm aware of that were just released at recent conferences highlighting this technology and all the various applications that it could be used for. And then also I'd like to jump in a little bit after that into a discussion of digital PCR. Digital PCR is something that's gaining a lot of interest around the world right now because it's different, very different from qPCR. And a lot of times where qPCR stops or reaches a limit in what it can do, then digital PCR is another technique that can actually be employed to get a more refined answer. And so I'd like to talk about digital PCR as well, because this platform is not only a qPCR system, but also a digital PCR system. And then finally, I'd like to introduce a little bit about our discussion about our somatic mutation detection assays. This is new chemistry, which not only can be used on traditional plates, like a 96-volt plate or 384-volt plate in single tube format, but it can also be used, and a lot of researchers are doing this now, combining that with OpenArray technology to really get a very refined look at rare variants. And then the last bullet point, which you don't see because I forgot it, and it's the most important bullet point, is really the key question and answer session. So I hope I can spur some discussion. We have plenty of time before lunch, and without further ado, let me launch forward into TaqMan chemistry. So a brief overview again for those that are not familiar. This is technically called the 5' exonuclease assay. We refer to this as TaqMan. Everybody in the company calls it TaqMan now, and everybody in the industry and the research community calls it TaqMan. And does anyone know where the name TaqMan came from? Anybody wonder? I mean, Taq is Taq polymerase, right? Taq polymerase enzyme. But what about man? Think of the video game, Pac-Man, right? Pac-Man's going through and he's chewing, he's eating all the little points. That's not a joke. This is literally where the name TaqMan came from. Because what's happening is the 5' to 3' exonuclease activity of the enzyme is encountering primers in its path and basically chewing it up, digesting. So if we start with the forward primer over here, and then the polymerase is moving and encounters the TACMAN probe, which is short oligonucleotide, has a fluorescent label on the 5' end, has a quencher molecule on the 3' end. As the polymerase encounters that probe, it just simply digests it. And so when I digest that probe, we liberate fluorescence. So this fluorescence that you see that's liberated in solution, it's separated away from the quencher, and then what happens is we get a rise in fluorescence level. So at every PCR cycle, we're doubling the amount of fluorescent molecules that are in solution, and so we're getting an increase. We get this sigmoidal increase as we go through PCR cycles. And so, of course, the data that we collect then is at this point right here. You just draw a threshold. And that's basically how it works. Now, this is the case for, there's variations of this, right? This is an example of a gene expression assay. If you're, for example, a genotyper and you're looking at biallelic determinations of a polymorphism, you're basically going to use a SNP TACMAN assay, which looks exactly like this. The only addition then is that we have an extra primer in there. So we have one primer that's specific for one allele that has let's say the FAM fluorophore, and then there's another oligonucleotide in there which is identical but just different by a single base. It's complementary to the other allele, and that has a different fluorophore. So that's all mixed together, and that's what we actually have inside a TACMAN assay in that single tube. And that's in addition to that, that's the physical part of it. But in addition to that, we actually have a lot more built in behind that. And so earlier Tony was talking about the TACMAN assays and applied biosystems, the experience we've had in Life Technologies incorporating all these different great brands. One of the great things that we inherited from applied biosystems is the expertise with our bioinformaticists, right? So we've been in the business at the very beginning when qPCR first came out. So we have literally over a decade of experience with our bioinformaticists actually designing successful assays. So when you actually look at a single tube and you buy it from Life Technologies, what happens is that we went through a very large bioinformatic gauntlet. So we basically look at SNP masking, repeat masking. We apply algorithms based on our experience of designing assays for 10 years. So these computer algorithms basically can predict what is going to be a successful assay that's going to work in your hands. So there's a lot of this actually that's involved and that the researcher doesn't necessarily see. And the important part of this message is that you don't have to do all of this, right? This was actually already done by us in silico. In many cases, these assays have been wet tested. For example, all human SNP assays, when they go out the door, they've literally been functionally tested. So a bench scientist in our labs has actually physically tested it against a selection of human DNA samples to ensure that we're actually getting performance. If it doesn't pass performance, then we don't release that assay. We notify a customer and say, sorry, we're going to redesign this. So there's actually a lot built in to what's in that tube itself. Now, one thing is that's great. We went through all this effort to make great assays, but we have to make this easy for the researchers, for you to actually find this on the website. And so what we do is, we have actually a collaboration with NCBI where any genome that's fully annotated, basically the information for it is linked on our website, and you can graphically, in this case, zoom in at the chromosome level. You can zoom in on the map and just scale in closer and closer. And then what you can do is you can actually see the position of the actual assays for a given locus. And all these assays are reported down here. Now, the reason why this is important is because we want to make this simple and easy as possible. So if you're buying just a single assay for a single polymorphism or a transcript, you can just get that, or you can take this content and just easily populate it into different formats, into pre-plated formats. So that said about TACMAN assays, let's move on to a discussion of what does a researcher want to do if they want to look at not just a single marker or dozens of markers. What if they want to look at hundreds of markers or potentially even thousands of markers, and they want to use qPCR? There's always been this huge gap in this area here. What you're seeing on the y-axis is basically the marker content. How many genes or how many features are you looking at? Obviously, at the very, very high end, to get a full hypothesis-free look at a sample, basically, next generation sequencing is going to achieve that for you. And that's what we have depicted on the top there. On the x-axis, going across, that's the number of samples that could be interrogated by a researcher. And if you get into areas of where you're trying to screen thousands of samples, tens of thousands, maybe potentially even hundreds of thousands of samples, think about, for example, if you're doing a genome-wide association study, right? So you have patient cohorts of 10,000, 100,000 even in these studies. So how can a researcher actually embark in this study by trying to use TaqMan chemistry, but screen that many samples, or screen larger numbers of markers? There hasn't really been a platform to sit in this space, in this big space here, and in this space over here until now. So that's kind of what we call mid-density. So when we talk about on the side the number of markers, mid-density is considered this area here. High density obviously is here. This is an image of a microarray chip. You guys are all familiar with microarrays. Microarrays can have like hundreds of thousands or even like millions of probes attached to the array. But what if the researcher doesn't need that? Right? You've already done your initial studies with microarrays, and you need to go back and do more focused validation. So instead of looking at an entire transcriptome, you've identified the targets of interest. Let's say you're studying breast cancer, and let's say there's 100 targets that you'd like to look at. Why would you go back and continue to interrogate your samples using a whole transcriptome approach? Because you've already determined that 99.9% of the content of the array is uninformative. It's of no interest to you. So instead what you're doing is you're picking a smaller subset and saying, I would like to interrogate these further through qPCR. The limit in the past has been cost. TaqMan chemistry, obviously, it's not necessarily cheap. If you want to do qPCR with large numbers of markers or large numbers of samples, the cost gets prohibitive. So researchers have been limited to, instead of taking 200 markers, saying, okay, well, let's just pick maybe 10 or 15 that we can go after. So now with the advent of mid-density technology, and I'm showing you an image of an array there, the array itself physically, I don't have any to hand out, but I'm holding one right here in my hand. It's just basically the size of a microscope slide. So this array technology then would enable us to actually tackle both what we see here on the y-axis and the x-axis. So because of that, what happened is Life Technologies acquired open array technology. We actually bought a company called Biotrove in early 2010. So this actually is a much more mature platform. The Biotrove system has been out in the market, I believe, since early 2006. And when Applied Biosystems at the time, before the merger with Life Technologies, what happened is that they recognized the potential for the system for high throughput genotyping. So our first customers really to adopt this open array technology were all genotypers, because they were looking at larger numbers of markers and huge population samples. So what happened is initially we adopted this for our SNP genotypers. We said, okay, the array itself is very simple. If I describe it, inside that array there's 3,072 through holes. So that a single array is the equivalent of 32 96-well plates. So this enables you to do high throughput. This actually enables you to get the cost down dramatically, because imagine the cost of a qPCR assay if you're doing, let's say, I don't know, a 20 microliter assay or even a 10 microliter assay. Let's compare that cost to what it would cost doing it inside one of these wells, which is actually a 33 nanoliter reaction. And so the cost savings that we see is actually due to the fact that we're using so much less master mix. Right? The master mix contains a polymerase and the DNTPs. So we're using so little master mix in these reactions, that's why the cost goes down dramatically. So we get cost benefits, sample throughput benefits, and also what we do is that we position inside all of these arrays, we already pre-spot your assays. So that's another advantage, because imagine if you're working with hundreds of plates, a multi-month project. Maybe you have multiple labs cooperating together on a project, and now you're asking someone to go into the freezer and pull out of the freezer, like 50 tubes, 100 tubes, 200 tubes, and you're trusting that researcher that when they're pipetting manually over and over and over again, that they're actually doing this without switching the assays. So think about the possibility of human error. So the other advantage here is that when we take these arrays, we take your content. You go to our website, as I showed you on that screenshot before. You select the markers that you're interested in, and then we take that content and we pre-spot these arrays for you. So the assays are already fixed in position. So you're not adding the assays at this point. To describe the array a little bit more in general, how the sample is actually introduced in there is through a very simple process. It's through capillary action. So what happens is the surface of the array is hydrophobically treated. The interior of the through holes, all 3,072, they're hydrophilically treated. So what happens is when the sample then gets introduced into the array, it wicks the sample in. And I have a couple slides demonstrating that a little further. So this was the technology. This was the original system that we're showing you here. It's called the NT cycler, open array NT cycler. And the array itself, there's different formats. This is just an example here of, let's say gene expression formats. So, if the numbers, that you're seeing on the bottom of the array basically mean the number of markers that you're interrogating per panel times the number of samples that you can look at on a single array. And so as an example here on the far right, we're looking at 224 different TACMAN assay gene expression markers on a single array, and they're all spotted sequentially in 4 squares, and that enables you to actually look at 12 samples simultaneously. So imagine trying to go to the freezer, take out 224 single tubes, right, and do this by hand. So it really simplifies the process. Of course, if you're not looking at large numbers of markers like this, you can get down to as few as 18 markers. We spot the 18 markers in triplicate, so you can quickly see by color, where we actually introduce all the samples on the array. Now, this is just an example of what we have currently. There have been many requests from researchers. They say, well, okay, this is great, but I have more than 224 markers, or I don't really have this many. I want to do lower than this. So we do have a custom process. We can kind of work with researchers if you don't fit, within the the inventoried formats that we have. If you're a genotyper, can I just see a raise of hands? Who's involved in genotyping, SNP genotyping? Okay. Okay, there's more hands going up. Okay, great. So if you're genotyping and you're interested in spotting our SNP assays onto these arrays, the formats are very similar. They're slightly different in that we actually enable you to add multiple samples per square. These squares we actually call subarrays, and I'll explain that a little bit further in upcoming slides. But we enable you to actually go as few as 16 SNP markers, and if that's the case, you can interrogate 144 samples in a single array. You can run multiple arrays per day. You can literally process thousands, tens of thousands of data points in a single day, as opposed to months. Those were the custom panels for gene expression and genotyping. This is where you're picking your content. You're telling us what you want. Now, what we also have that I think will be of interest to a lot of researchers are what we call these pathway panels. Curiously, what else is out there that I'm missing that's involved in this entire cancer pathway, let's say? Instead of just looking at the 10 or 12 that I think are interesting from information I got from doing research, let's just see what else is out there. What are other researchers looking at? Let's see if we can find any other connections. So we took that idea and said, okay, the open array layout is ideal for this because we can actually make these pathway panels even larger. So what we did, and we have these already available, and we'll be adding more to this list, but here's a listing of the human kinome panel. We have a human cancer panel, inflammation, stem cell panel has been of interest to a lot of researchers. The stem cell panel is a collection of 600 something markers basically for differentiation and pluripotency. So it was really helping a lot of researchers screen their stem cells for stemness, trying to characterize induced pluripotent cell lines, embryonic cells. And in addition to that, we have signal transduction panels as well. With these inventory panels, what we have is a much larger format. So the array itself is physically the same, but what we did is, obviously it's larger than the 224 that I showed you before. In order to fit 600, 700, 800 of these markers on an entire panel, we fill up an entire square, like an entire section of the array. So in this case, like 25% of the array is devoted to a single sample. So you could basically interrogate, just a few samples, against a large number of markers. That's one comment we've gotten a lot from early adopters of this technology, is they said, this technology is great, but the problem is it doesn't fit my numbers. I don't have, I can't meet your minimum order requirements for a custom panel. You're asking me to order at least 10 of these arrays. Well, I don't have that many samples. I only have 50 samples. You're asking me to order, 200 samples worth. And so what this does with these inventory panels, it allowed researchers now to just order these individually. You just get a single, 2, 3, 4, 5, whatever you need. So we'll actually be adding considerably to this list. We just released these, about 6, 7 months ago. We'll be adding quite a bit more as the months go on. I'm depicting down here a genetic barcoding panel. This is a collaboration we did with University of Miami, where they discovered that with as few as 32 human SNPs, we could come up with a genetic barcode that could identify a patient sample. So we can not only determine the sex, but we can actually identify that, okay, this sample is literally you, right? And then they also have like 64 and then 32 SNP panels as well. So these were actually released, and I think these would be of tremendous interest to anyone that's involved in biorepository work, sample tracking of large patient cohorts, trying to go back and validate and say, okay, we know the sample is patient 654, 6 on the list, because we confirm it with the SNP genetic barcode. In addition to that, is anyone engaged in microRNA studies, microRNA profiling in either human or, not microarray, but microRNA? Well, if you were, we have microRNA panels available on the OpenArray as well. And the reason why we thought this would be an ideal fit for OpenArray is that previously, when researchers were trying to profile our entire microRNA library against our samples, we have a collection of 750 human microRNA and rodent microRNA markers. What they would do is they would use what I'm depicting here as microfluidic cards. Microfluidic cards have been used, I believe, for about 6 or 7 years by our customers. These are 3D 4-well plates. They were very successful early on because we also did the same approach. We actually took the content, the TACMAN assays, and we spotted them inside the 384 wells. So they were completely contained. But the problem researchers had with this when they're doing microRNA studies is that they would have to run too many of these cards in order to complete this project. And so the workflow was kind of tedious, and if you had a smaller project size, that would be fine. But if researchers were literally looking at dozens, hundreds of targets, we thought instead of running multiple cards, we'll just spot the content because of the throughput capabilities onto the array itself. So we've been doing very well with the microRNA panels. Those were the assays that were spotted. Those are the different formats, right? I explained that you don't actually pipette manually the assays into the arrays. Now, what's missing from the reaction itself now? We have the assays in place. We're missing the sample, and we're missing the master mix. And we don't expect you, of course, to manually pipette that by hand into the little arrays either. As far as I know, there's no multi-channel pipetters that can allow you to pipette 3,072 wells. So what we have is we have an automated loading device. This is called the AccuFill system. That's what I have depicted here in the top. The AccuFill device is it's a liquid handling deck. It's very simple. It comes along with the instrument. You just lift up the door. And then there's a picture here of what's inside the loading system. And it's essentially just you're looking at 4 arrays, 4 of the open arrays lined up in position. There's a 3D 4-well plate right here. It's hard to see because the plate is black. But there's a 3D 4-well plate. That plate actually contains your genomic DNA or your cDNA that's been prepared, and it's already mixed with the master mix. So that's already in the 3D 4-well plate, and you just position that 3D 4-well plate inside the instrument. And so all this machine is doing is coming over, grabbing the pipette tips. It's grabbing 48 tips at a time, grabbing your samples, and then is moving over here to one of the arrays. And then here's a zoomed-in image of what's happening. There's 48 tips that are coming down together, and each of those tips is making contact with one of the subarrays. So if you think back to the colored schematic, where you saw the different color blocks, each block is being, the sample is being introduced one pipette at a time into the block. And if I zoom in here, this here is actually just one of those squares. There's if you zoom in, this image again I showed earlier, but if you zoom in, there's 48 of these squares on the array itself. Within each of those squares, there are 64 reactions. So 64 times 48, that's 3,072. What's happening is when the pipette tip comes down, it actually makes contact with the corner of one of those squares, and then just simply glides in a serpentine fashion. It just goes back and forth and just introduces 33 nanoliters, 33 nanoliters, 33 nanoliters. And the loading is actually very precise. Because it's capillary action, the volume that's actually being introduced into the reactions is determined by the shape of the through hole. And so since the shape of the through hole defines that volume, we're getting very precise loading of 33 nanoliters plus or minus 1 nanoliter. So the loading is very precise and it's automated. And the loading of the array takes this instrument about a minute. So if you think back again, okay, how long would it have taken me to set up 32 96-well plates by hand, manually pipetting with a multi-channel pipetter, and how many sources of error, how many points of error could have been introduced, then you really realize the potential of a technology like this. What I'm showing you here, in these two images is, remember I said for genotyping, you could actually spot multiple samples per subarray. So what happens then is the instrument would just discard the pipette tips. It will go back and grab more pipette tips, go into different sample wells, and then come back in and just reload the other half of the subarray. So that way, instead of just having 48 samples on an array, now all of a sudden you're looking at 96 samples per array. If you're going through this format right here, then basically you can look at as many as 144 samples. So the loading is very simple. And that's basically all that comprises the reaction, right? The assay was already in position. We added the master mix with your sample, and that was done by this automated loading device. So there's really no hands-on introduction for setting up this workflow. So I want to segue in and lead into this new QuantStudio 12K Flex platform. So the reason why we came out with this technology or this single system is that before this instrument was released, what we had was different formats, right, that were available. The TACMAC chemistry, was available through single tubes. I explained the 96-well plates were available. These are the fluidic cards. These are those 3D 4-well fluidic cards. All of these could be run on, let's say, the 7900 real-time instrument. I don't know if you guys have those in your lab. This is the VS7. This is a newer real-time instrument that we have. This instrument was very popular because of the flexibility, right? You can actually see I'm showing multiple thermal cycling blocks next to this instrument. And the reason why it was popular is because a researcher can easily switch the blocks. So now you have a system that's very versatile. You don't have to buy a system and then later when the project scope increases, then have to adopt another platform. Now all you have to do is just simply place the block in there. And it takes about 30 seconds. So the limit, though, was that this system, this is called a VS7, was that the system could not do even higher mid-density sample throughput. It could not run the open arrays. In order to run the open arrays, we had a dedicated system, which was the older open array NT cycler right here. So the idea, and of course this system, and I was explaining this to researchers outside, this system can only run open arrays. So what happened is that this platform, if it's in a lab where this is all you're just doing high throughput research all the time, you're screening large numbers of samples, well then that's fine. But the majority of researchers basically didn't have projects of that size all the time. So what would happen is if you look out in the market for mid-density, high sample throughput platforms, us and competitors, what would happen is that you'd see platforms that cost, in US dollars, $200,000, $240,000. And then a researcher would say, okay, well, we bought this platform, we got the grant money, and wow, this thing has collected dust now for about 4 or 5 months, 6 months, or even a year. You get excited, you buy the platform, you run a project, and then the project's finished, right, in a couple days. And then the rest of the time, this machine's collecting dust. So, people saw that as a problem. We saw this as a problem right away. So the idea behind this is that let's go ahead and launch into a new platform which can run all of those formats. So we can say that this is our, a universal platform. This is our newest, in the line of qPCR instruments. It's the latest development after, 10, 12 years of qPCR instrument engineering and research, that we can release something that is truly unique in the market. There's no other competitor that can say that they have something they can do everything from a single tube all the way to high throughput 3,072 reactions, right? So this is an image of the new QuantStudio. We actually announced this at the end of 2011 at ASHG. So we announced this. It was a big splash, marketing campaign to release this. And these instruments have actually been going out and selling now since the beginning of the year. Clicker died. Uh-oh. Okay. I don't want to bore you too much with like all the little details of the instrument, but just briefly, let me back up real quick. If you look at the front of the instrument, and if you saw the actual instrument that was out there in the lobby, you'll notice that there's an LED screen, LCD screen on the front. And what was really nice about this is that you could actually look at your runs in real time. And so you could see this up on the touch screen. So you could control the instrument through the touch screen without a computer. You can run it with a computer. You can run it without the computer. You can monitor the runs remotely. You could actually see the amplification plots on the screen itself. So we just basically took all the comments and the features that we saw, like in the progression of real-time instrumentation over the last decade, and try to put everything into this platform that researchers said they wanted. So like I said, the instrument itself can run without a computer. So what we have is we have onboard storage on the system. So you can actually store up to hundreds of runs on the system. There's a flash drive on there where you can store the data and get it off there later. You can actually program experimental files, put that information on a USB stick, and then you can go in and just plug it into the instrument and run it like that. So a lot of versatility. And with the system also, you can basically set up a run, and then you can just go home. And then you can have the machine email you the data to your house. So you can stay up late at night in front of your computer looking at the data that was generated without waiting and sitting by the machine the whole time. One of the things that we found out right away that was needed, it was the illumination source. So there's a lot of different real-time instruments out there that use different illumination strategies. Of course, even within Life Technologies, if you look at all the instruments that Tony introduced earlier in the presentation, we use a variety of light sources. We use focus LED, focus wavelength LED lights. We use broad spectrum halogen bulbs. So what we figured out actually is that there's a new generation of super bright LED lights that are out there now. So these are the same LED lights that you're starting to see in like football stadiums, right? So these are really super bright uniform lights. And this brighter LED basically allowed us to have the resolution of not only looking at, let's say a single assay or single well, but also going all the way up to the open array itself. So we took this LED technology, incorporated it into this new system, and then with the LED, we're actually able to illuminate the fluorophores that are of most interest to us. The FAM and the VIC fluorophore, of course, these are FAM's it's used in all of our TaqMan assays for gene expression, microRNA profiling. FAM and VIC are used if you're doing biallelic discrimination using our SNP assays. But in addition to, and you'll see here that the LED spectrum is actually here in blue as compared to the halogen bulb, which is used in other systems. It's longer wavelength. So it wouldn't really excite the FAM and VIC fluorophores as well in these tiny 33 nanoliter reactions. We found that while it was sufficient to illuminate something in a 3D 4-well plate, it just simply didn't work when you're getting into nanoliter volumes. It didn't work as well. We needed to improve the sensitivity of the system. So we achieved that through this next generation super bright LED system. Now, that's FAM and VIC, but what about researchers that are looking at longer wavelength dyes? Are you sacrificing anything by not having that halogen bulb? And the answer is no. This system is a multiplexing system. It can actually interrogate dyes at short wavelength, 480 nanometers all the way up to 630, 700 nanometers. So if you're looking at longer wavelength dyes, if you're truly interested in multiplexing targets, you like to look at 3 or 4, let's say, viral assays simultaneously within the same reaction, and you want to go through the work of optimizing and trying to get the primers to actually cooperate and work together, well, then that's fine. The instrument is set up already to enable multiplexing. You can look at as many as 5 targets simultaneously. And the way we achieve this is actually, from an engineering standpoint, is because we use decoupled excitation and emission filters. So using these filters together with that super bright LED, we're actually able to illuminate dyes of all wavelengths and then accommodate all the different formats on the system. So it's a combination of those two that enable us to do that. And just an example here, just showing you a true multiplexing run. If you're looking at 5 targets, we have 5 genes that we're looking at with assays, all labeled with different fluorophores. What you're seeing in the light blue, this low amplification plot, obviously this wasn't excited as well because this was longer wavelength. However, we got good data, right? We got excellent data because we don't care about the actual amplitude of that plot. That's irrelevant because we know in real-time PCR, the data actually is all derived within the exponential phase of amplification. So we got perfect data from a 5-plex experiment here. So when we announce or release a new instrument like this, we of course have to do internal validations. We have to convince researchers that not only does our instrument say what we say it does, we have to demonstrate it by benchmarking it, comparing it to our other systems, comparing across all block types. We've done a lot of internal R&D studies showing this data, demonstrating not only is this machine as good, but it's actually better. So just a brief overview. Again, a reminder that this is not a niche platform. It's not just a system that runs high throughput open array. This is a system that will run 96 well, 3D4 well, TACMAN array. These are the microfluidic blocks, TACMAN array card blocks, and it does it equally well. So we just do this is standard. All vendors in the field will do this. They'll take a sample, like let's say a plasmid, and then they'll do a 10-fold dilution series of that. And the point of this is to demonstrate full dynamic range of the platform. So by doing this, we just took plasmid DNA, and we basically just went all the way from low copy numbers, as few as like 10 or 5 copies, and we do a 10-fold increase across to show you that we're actually getting reproducible amplification and that the PCR efficiency is constant throughout that entire dynamic range. Now, this is data of the actual open array itself, and the reason why I show this is because there's a lot of confusion sometimes with interpreting CT values. So if you look at a cycle threshold value on a traditional real-time system, for those of you that have done a lot of qPCR, we know that typically if you're talking about a single copy, mathematically a single copy of a template would probably occur at around 36, 37, 38 PCR cycles. Before it actually gives a CT signal. That, of course, is dependent on what the assay is, what your what's actually in the master mix, what the rise is above baseline fluorescence, and also where you set the threshold to determine that CT value. And the CT value is what you're seeing here depicted in the y-axis. So the confusion arises from the fact that when you move into nanoliter size reactions, those numbers actually shift dramatically. So if you tell a customer, okay, a single copy of in a well is interrogating and demonstrating a CT value of, let's say 37 on a traditional real-time system, you're not going to see the same thing in a nanoliter environment because the volumes are much smaller and you have a much smaller window of amplification rise above the baseline. And so what happens is the CT, if you think it in terms of molecules per volume, the CT number is actually shifted dramatically to the left. So what would be 35 cycles, let's say, in a traditional real-time system would actually occur at around 25 cycles. So that's one thing to consider, and that's no big deal because remember the CT value itself is an arbitrary number. There's no meaning in that CT value unless you actually compare it relatively to another reference sample or a reference marker. So everything is shifted dramatically. So what we do here is we just demonstrate that yes, even with the open array, we're able to achieve a very good dynamic range. We cannot match what we showed in the previous slide, which is 9 orders of magnitude, right? It's just simply, it's not a limitation of the machine, it's a limitation of the volume of the reaction itself. We just simply can't exceed 7 logs here. And of course, what's happening in this instance, this is the lowest dilution point. This is probably at around, let's say, 100 copies, 50 copies of target input. While you could actually get very consistent CT values using a 20 microliter reaction, when you get into nanoliter reaction environments, what happens is we run into a problem that is independent of the machine itself, It's Poisson distribution. So it's just simply Poisson. We can't control the number of copies that are reproducibly being introduced into nanoliter reactions. Because what's happening is, obviously, you're just getting 50 copies in there once, and then in the replica, you're probably adding maybe 20. It's bouncing around quite a bit. And that's what you're seeing here. So just to back up again, if we look at the overall workflow of OpenArray on the system, what happens basically outside of your hands is this, right? This is what we do at Life Technologies. You take the assays like I described. We actually these are the same assays that we would manufacture as single tubes. It goes through the same design process. We take all these assays, let's say hundreds of markers that you're interested in. We spot those onto the arrays. So when the researcher receives the array out of the freezer, it has your content. This part down here is actually what you're doing in the lab. This is the actual loading part of the arrays, and also the actual run, and then the analysis of the data. Once you actually load the array with that system, the only hands-on part here is that you're simply clamping the array itself. You have to contain the array, because remember, the array is open, right? We call those in through holes because you can literally look through the array. And we call these open arrays because you can literally look, if you hold it up to the light, you can see straight through. We have punctured 3,072 holes in there. So how do you actually contain this 33 nanoliter reaction in there? Well, and how do you actually thermal cycle? Can you imagine going up to 95 degrees with this open array with only 33 nanoliters of fluid? What would happen literally within the first couple seconds is you just evaporate everything in that reaction. So what we have to do is we have to take the array and seal it inside a cassette. So it's a glass case. The glass case then is filled with immersion fluid. This immersion fluid is similar to mineral oil. It's not mineral oil, but it's similar. But we fill up that case with liquid solution. This is not aqueous. It's non-aqueous. It's an oil. So what happens is now you have the array with the aqueous reactions. It's sandwiched between two layers of oil. So you have oil on top, oil on the bottom. So of course, what happens then is the entire case, along with the glass, the immersion oil, the stainless steel array, the assay, all of it gets thermal cycled up and down together. And so because of that, we get asked a lot, okay, what are the cycling times? Can I actually run a really fast qPCR reaction like I would traditionally on a regular instrument? Can I do 40 cycles of PCR in 35 minutes or 45 minutes? The answer is no, because a thermal transfer going across the glass to the oil into the stainless steel array just takes longer, so the cycling times are a little bit longer. Not much longer. For gene expression runs, our cycling is about 2 hours and 10 minutes, so it's still not really that long. For genotyping, we like to actually go a little bit further, optimize our protocols for 4 hours for genotyping runs. But what happens is after you've actually loaded the oil into the glass case itself, you just take the glass case and you can load up to 4 of these arrays into the instrument. And that's what you're seeing down here in the bottom right. There's the side arm of the machine is opening, and you can load a single array. Remember, this is 3,072 reactions. Or you can load 4 arrays. Now you're up to 12,000 reactions. So now if you think of the name QuantStudio 12K, 12,000, where did this number come from? It came from the fact that we can actually run 4 of these arrays simultaneously. You can get 12,000 data points of qPCR in a single run within 2 hours and 10 minutes. So in 2 hours and 10 minutes, you could accomplish the equivalent of maybe 2 or 3 months worth of work. This illustrates a little bit better, just showing you 384-well plates that are stacked up here. The single array, if you're doing gene expression, mathematically, whoever's quick in math, you'll say, okay, 3,072. That's not equal to 7 384-well plates. That's actually equal to 8 384-well plates. All right, that's true. But for gene expression, there's certain through holes that we actually don't use. We don't spot assays into them. And so mathematically, for gene expression, it breaks down to 7 of these 384-well plates. Or you could look at it as 28 96-well plates are the equivalent of a single one of these arrays. A lot of throughput. The throughput is achieved, it's illustrated better, I think, here. So if we say this is a single instrument, this is a single researcher running this project, with one machine, basically you could come in at 9 o'clock, basically load up 4 of these arrays. Remember that the green section of this graph, right? This is the machine doing the cycling. You're not doing anything at this point. You're on the internet, Googling things. So basically what happens is the workflow is just this part right here. It's just 15, 20 minutes of preparing the array, setting up the machine, so forth. So there's a lot of downtime where the machine's doing all the work. So that's why this is a very easy and doable workflow for a single person. A lot of times we get accused of presenting something on screen that's not realistically achievable in the lab. We got someone that's experienced for a year doing something, and this is what we can do. Now you go do it. But in reality, the workflow is so simple that this really is achievable. So a single person runs 16 arrays per day. That's 43,000 data points if you're doing qPCR. If you're doing genotyping, just look at the top half of the screen first. If you're genotyping, what happens is that you can actually, because the run times are longer, you run 4 at a time. You can basically run 12 open array plates. That's 36,000 genotypes that you could interrogate in a single day. Now, for even high throughput genotypers, if you want to increase this further, you say, 36,000, that's not enough for me. I need more than this. Well, if that's the case, then we have these offline. This is just our standard 9700 thermal cycler. I've had these out for a number of years. Researchers can actually cycle these arrays offline, so off the machine itself. Because remember, for genotyping, we're not really capturing qPCR data in real time. Right? Traditional genotypers, you just amplify, and then you do endpoint detection. Right? So you don't need the machine to be using detecting fluorescence at every cycle. So that allows you to actually run up to 8 arrays in a single 9700, just do regular PCR, and then when you're finished, take those arrays and put them into the instrument and just read. Use the machine as a reader, fluorescence reader. And it only takes 5 minutes to read an array. So I'm just showing you an example here that, if, let's say you had 2 of these 9700 sitting there in the lab, you can run 16 at a time. So that means a single researcher with very little hands-on could run 32 of these arrays in a genotyping setting. And that's almost 100,000 reactions. So these are, realistic numbers. Is anyone impressed yet? And for even higher throughput. So you're saying, okay, you know what, that's great, but I really have higher throughput needs. I work for an ag bio company, and we're screening seed lots. And you know what, I have to screen tens of thousands of seeds on a weekly basis against hundreds of SNP markers. Everything you showed me is great, but I need even higher throughput. So we have this twister arm. It's a robotic device that sits next to the machine. What this allows you to do is stack a whole bunch of plates together, and then the machine just automatically grabs a plate, puts it in the machine, does the run, takes the plate out, grabs another one. So it just does this all night long. So you don't even have to come back to work the next day. You can stay at home because the machine will send you the data, right? It emails the data to your house. So this is even higher throughput. And we calculated if you're doing genotyping, for example, you can actually get up to 2 million data points, 2 million genotypes if you stack up. You can stack up to 55 of these, times 4. So hundreds of arrays. Let's talk a little bit about the software features. This is very important, because I talked about the hardware, what's so great about, the way the machine works. All that's pointless if you can't make a machine user-friendly. You have to make it easy for the customer to interpret and understand. Admittedly, some of our older instruments were a little, hard to decipher in terms of the software. So we really worked on eliminating that bottleneck for the researcher. So we came out with a software platform that should be intuitive. Let's say our field application scientists came and trained you, and then you were distracted on other projects for a while, and then you come back 2 months later, you forgot everything that you learned in the training. Well, we designed software so that someone could come up and just figure it out on their own. Worst case scenario, you pick up the phone, call tech support, and just, hey, walk me through this. But it should be very easy to understand and very easy to figure out. You shouldn't have to have someone come back out and retrain you, because that's a big bottleneck to your research. It slows you down. So basically, we have this new software that basically had a lot of users test, provide us feedback. We allowed the ability to actually have multiple software packages for gene expression, digital PCR, genotyping, high-resolution melt analysis. All this stuff were separate software packages, but we said we have to make this simple. We have to incorporate this all into one software framework. So we actually have all these enabled in the software itself, and it's actually built in. So this is just basically the basics of the software for the instrument, and then all those different software packages that I just showed you in the previous slide, all those can actually be automatically incorporated and built into the software. So you don't have to close a screen and go somewhere else on the desktop and look for another program or export data, move things back and forth. It's actually all built into the same framework. And as an example here, this is just the Expression Suite software that's built in, very easily accessible. Expression Suite software, the really nice features of this are that we had a lot of tools built in for correlation plots, volcano plots, looking at p-values and correlations, heat maps, basically for screen. This is very important because imagine when you're looking at let's say hundreds of targets simultaneously, right, across dozens of samples. Are you going to one by one click your mouse and look at the amplification plot one by one by one? Right? That gets incredibly tedious. The whole point of looking at large sets of biomarkers is to actually get a profile. Right? So we have heat maps built into the software where you can just do a very quick heat map analysis and allow the software to actually segregate all your samples. For genotypers, the genotyping software is also built in. Oh, by the way, so this software, the previous software, this is free software. So this is downloadable off of our website. And the nice thing is that, yes, the software, is part of the QuantStudio instrument package. But if you don't have the QuantStudio system, but you have another applied biosystems system already in your lab, you can take full advantage of the software. This is free software. So gene expression is downloadable. You can actually look at all the data from your other applied biosystems or Life Technologies instruments that you have in your lab and analyze the data using our newest, state-of-the-art, analysis package. And this is free software for everyone to download off the web. What we can't help you with is if you're using a Bio-Rad machine or Roche or somebody else. There's workarounds, but we won't go out of our way to help you too much. This is an important feature because this was actually a customer of mine back in San Diego. It was a biotech company that had been using our 7900 instrument for years. And they said, and they're big genotypers. And they said one of the limitations that they saw was that when doing traditional genotyping, remember I described that genotypers just look at endpoint data, right? They thermal cycle, and they basically just arrive at an allelic discrimination plot, a cluster plot, or a Cartesian plot. So what happens is basically these are obviously no template controls, and you're getting two population groups shown up here, right? So you get a group in green, and then you have a group in red that are showing up as two populations. Now the problem with this is that if you look closer into the green cluster, you'll actually see two separate subclusters, very distinct subclusters. However, the algorithm, the software is actually defining that as a single population, allelic population, because of the proximity. What this new QuantStudio system enables you to do is actually optional, if you want, to do genotyping with real-time capability built into the run. So not only would you just run it and then just image at the end, you have the option of actually looking at fluorescence at every cycle. So then this allows you to go back and trace the fluorescence as it migrates backwards in time. So what this allows you to do is you don't have to go back and rerun the experiment and say, okay, well, we tried 40 cycles this time, let's go to 35 cycles, or let's try 30 cycles. A lot of different assays perform optimally at different PCR cycles. Some have to go all the way to 50. So what this new software allows you to do is just run it once, do 50 cycles on all of your different SNP assays, and then instead of physically going back and reproducing the data and just modifying it, all you're doing is you're marching backwards in the software itself because the data was already collected. So if you see the traces, the algorithm, and the user can actually go back and manually call this as well. They can go back and clearly see that this original cluster, which appeared to be a single allelic population, actually migrated completely differently. And so this is the end result. You identify this as a completely different group. Obviously, we would expect if there was a true homozygous population for one allele, if it was really beautiful, you'd expect it to appear actually over here, right? But that's not the case with all assays. Some assays, they just tend to blend together a little bit just because you ran too many cycles. So this enables you to do that. So this was actually one of my customers in San Diego that really called for this, asked for this to be built into the new software. And since we incorporated this into the software, it's been incredible, the response from all the genotypers now. They see this and they're like, this is amazing. We never had the ability to do this before. I'll briefly just touch on applications. Now, obviously, I'm not testing your eyesight. I know you can't see this. What I'm attempting to do here is just show you some recent screenshots of posters that were presented either at AACR, ASHG. There's an upcoming American Society of Molecular Pathologists, AMP, that's coming up in October. So I'll show you a poster there. But this is like early work here where basically with our researchers, groups were just studying, you're studying the use of open array technology for various applications. So in this case, we're basically looking at testing of biofilms, water sample testing. So the ability to actually screen very quickly water samples for different bacteria, pathogen particles located. And the reason why I'm not zooming in on all this is because there's nothing novel here. Remember, this system, although the novelty is that it's in this array format, it's a 33 nanoliter reaction. That's the end of the novelty, right? The rest of this is qPCR. So the way the data is interpreted and collected, it's still qPCR. So that's why I'm not going to bore you with all the details because this is no different for if someone had done this in a traditional real-time system. The power of this was the throughput, the ability to actually screen large numbers of samples quickly and get an answer very quickly. This is a poster that was presented at ASHG. This was basically our researchers at Carlsbad headquarters in California, where we set up this year a special stem cell applications lab. It's a lab where basically it's a dedicated team focused on all areas of stem cell research, from at the cellular level all the way to transcriptome profiling. So they obviously are adopting this technology as well, incorporating it into their labs and demonstrating data. So this has been, what they're trying to show here is that it allows stem cell researchers to go back to their cultures and just quickly characterize, even at different passages, their cultures. So they're looking at induced pluripotent stem cells, embryonic stem cells, and just using this open array technology as a screening tool, basically to characterize. And what they did is they did PCA analysis, principal component analysis, to simply just compare the results. What does it look like with microarray analysis, which is a lot more cumbersome and tedious? Can we achieve the same answer using a very quick and fast qPCR methodology on the open array? And yes, they were. They basically achieved pretty much the same answer there. And again, I'm still testing your eyesight here with more posters. But this is a recent poster actually with a collaboration with the FDA back in the States. So with our company and the FDA, we're basically just using, the title is somewhat misleading. It says multiplex analysis of pathogens. So we're not actually multiplexing the assays within the wells. They're saying multiplex in the sense that we can do the equivalent of multiplexing, but basically do these as singleplex assays in a high throughput format. So basically enables us to, let's say, spatially multiplex, geographically multiplex on the array, without having to actually try to test all these different assays out and get them to work together. So the FDA always has this panel of like 30-something potential threatening pathogens, and that list is changing all the time with different strains and variants. But they said, okay, with the open array technology, we can quickly just come up with easy panels to detect all these pathogens and just use it as a screening panel for routine studies and analysis. And again, all this information here, which is so tiny that you can't see it, this is just standard qPCR results, so I'm not getting into that. And admit it, I can't even read this. This is work that a group did at basically North Shore Cancer Center in Hempstead, New York. It's a recent poster where they're looking at ovarian cancer. And so what they were trying to do here is basically show in ovarian cancer if they could come up with a predictive gene signature for survival. survival after surgery and chemotherapy. And what they found is they found a 90% correlation. Actually using this technology, they were looking at microRNA profiles in plasma. So if they could actually just look at the circulating microRNA levels in blood, could they actually come up with a predictive signature, gene expression, or microRNA signature predicting survival rate? And they were very successful in the early phases of the study using those microRNA panels that I showed you early in the talk. They used those, and they actually found in some cases, like MIR16 had an 80-fold increase in expression values for the population group that exhibited, like very high or full survival 5 years after surgery and chemotherapy. So, and there was a number of other mirrors as well that showed, 40-fold expression increases, 20-fold expression increases. So very, very powerful and quick to use screening tool. And they worked with us on this because one of the initial requirements for the protocol was that we required about 100 to 200 nanograms of starting RNA material. And so that's a lot, when you're asking, trying to interrogate small sample sizes, or you have, biopsied samples, or, you just basically have very little, let's say, fluid to work with. So we have, optimized a protocol where you can actually, using a preamplification strategy, you can use very, very, very low levels of input material and still preserve the expression relationship without bias. So, in previous poster, I showed you that there's a stem cell applications lab at Carlsbad. They've actually already demonstrated that we can get down to single cell gene expression detection levels by using this preamplification method. Preamplification method has been used and well established in the literature now. I'd say going back 5 or 6 years. So as long as you keep the PCR cycles to a minimum during the preamplification, you basically avoid the bias that gets introduced through exponential PCR. If you do too many cycles, of course, you will eventually always introduce bias. But we figured out a very clever way of minimizing that and preserving that gene signature. This poster, basically, although small, you can pretty much look at the middle and figure out what the message here is. It's correlation analysis, correlation plots, very high R-squared values. This is a group that I work with at Cedars-Sinai Medical Center in Los Angeles. And the reason they actually included me on this poster as an author, and they'll be publishing later this year as well. And the reason why I like showing this poster is because they were skeptics of this preamplification method. No, we don't want to do preamplification. We heard from others it introduces PCR bias. We don't want to do this. So we actually, convinced them by essentially doing the study for them for free. Basically by, they provided us samples, and there was 2 very important things they wanted to answer. One, do we trust the person at Life Technologies that said preamp will not introduce bias? So they tested that. And they also wanted to know, more importantly, can we compare FFPE, so formalin-fixed, paraffin-embedded samples, can we compare that back to their matched frozen, pristine samples and still arrive at the same signature? Do we get the same correlation? And to embark on this study, there was so many different comparisons that they did. They actually compared different reverse transcription methodologies. They used random priming, gene-specific priming, oligo-DT priming. They had us design special assays for the FFP material, because as you know, FFP samples are extremely degraded. If you use, large amplicons, the amplification efficiency drops down dramatically. So we designed very short amplicons for them. The end result of all of this is they saw a very, very high correlation between using small amplicon designs on their FFP sample set compared to the fresh frozen. That was very important for them. And then also they were able to show that with preamplification, they were able to use very little of their precious patient material. They could get away with using 10-fold, 100-fold less material using this amplification approach and still arrive at correlation, very high correlation to what they were expecting. So that was the message here. This is a very thorough analysis or a testing of the technology itself and our preamplification method. Let me jump off here a little bit and talk about digital PCR. And the reason why I want to talk about, first of all, who's familiar with digital PCR? Has anyone heard of digital? Nobody? Okay, so digital PCR is something relatively new, and it's something that's getting a lot of a lot of buzz, a lot of interest in the research community. And the funny thing is, digital PCR was first described in proceedings in 1990. The first paper describing digital PCR was back then. No one really adopted it. And no one thought it was something that was really usable. This is, okay, interesting paper, great. But it's not something we can actually use and apply in our lab and in our research. And so the reason why is because in order for digital PCR to be effective, you need a large number of replicates. So what digital PCR basically is taking a sample and basically taking that same sample. Now this sample has your material, your cDNA or your genomic DNA. It has your master mix. It has the assay of whatever target you're trying to interrogate. It has everything pre-mixed in there. All you're doing in digital PCR, you're saying, let's take this material and distribute it. Let's distribute it across as many wells as we possibly can. And so the idea behind that is that if we dilute the sample across enough wells, eventually what's going to happen is we're going to get single copies of that template by itself in that reaction vessel. So now we've isolated individual copies. So that's the entire principle behind digital PCR. And so what that enables us to do is to get an absolute measurement or an absolute count of how many copies of that target you have in your sample. So the obvious thought is, okay, if I'm trying to quantitate, let's say, viral copies, how many copies of HIV does this patient have circulating in their blood? Well, you want to get an absolute count of that. So in order to get an absolute count, we can partition copies into their own wells. And so if you think of Poisson distribution, Poisson math, right? So we get to a point where you dilute enough. How do you get to 1 copy as opposed to 5 copies or 10 copies? Well, you dilute enough, you're eventually going to get so many negative wells, like empty wells light up, and that's fine. That's fine. We actually want a large number of negative wells because when we have a large number of negatives, we know that we're getting into that Poisson range of distribution, and that we can reasonably be assured that the wells that do light up positive have a single copy in there. And so all the machine is doing at this point is just counting the number of wells that lit up on the array versus the number of wells that are not lit up. And from that number, that count, it's going back and inferring how many copies you have. That's the principle of digital PCR. And it's an extremely simple technique. Digital PCR can literally be done tomorrow by every single one of you in the lab using a 384-well plate or a 96-well plate. But as I said, the limitation there is that you don't have enough wells for this to be really effective, right? Because what's going to happen is, how do you know that if you take your sample and you split it across 96, that you really are going to get single copies in there, right? You actually may end up with 1,000 copies in a well, 1,000 copies in the next well. So you have to do a lot of work in pre-diluting your sample so that it actually fits into the dynamic range of whatever vessel or plate you're using, 96 wells or 384 wells. So in reality, digital PCR was just not very applicable, until now, when we've released these mid-density arrays, with the ability to actually distribute that sample across 3,000 wells, right? So you just gain an order of magnitude. And that's basically digital PCR, an overview description. The uses of digital PCR obviously is, viral quantitation, trying to ascertain true copy number. So if you're looking at copies of a gene and comparing patient A to patient B, and let's say, the resolution of qPCR stops at, let's say, like 2-fold discrimination. Let's say, you have 4 copies of a gene, an HIV susceptibility gene in one sample, or in one patient, and then you have 5 copies in another patient. That's a 20% difference. If you mathematically think back to a 20% difference in copies, how that would look in qPCR, the amplification plots would be right on top of each other, practically, right? Mathematically, I think they're 0.3 cycles apart. So you can't really, with confidence, say that there's a true 20% difference in copy levels. That's where digital PCR can actually come in. Studying like copy number variation at a much higher level. You can actually get down to about 10% differentiation. So if someone had 10 copies and then the other person had 9 copies of a gene, you'd never be able to do that with qPCR. But with digital PCR, it's been demonstrated. So in a lot of cases, we're seeing this pop up here as well. So researchers are looking at low transcript levels, low copies of transcript, and they're getting CT values that are coming up very, very late on their traditional qPCR system. And they're not really sure what that number means. Right? So something came up at 34. Well, we tried to reproduce that, and the replicates show up at either nothing or like at 30. What's going on here? We're running into Poisson mathematics again. So what happens is with digital PCR, you can actually take each individual sample and just get an accurate count, rather than just guessing. Okay, well we're plus or minus 2 cycles here, with a CT value of 34. So this is a very precise method of quantitation, or a technique for quantitation. To illustrate that, so you just kind of understand it better, this is an example of just taking a virus. Remember, this could be any template, really. I mean, this could be, let's say, DNA extracted from soybean. We're basically just trying to interrogate, any given locus. So what they did in this case is they just took an array and they did a serial dilution. They did a 2-fold serial dilution of this BK2 viral standard that we have. And basically, they wanted to be able to determine if they could get an accurate count of how many copies of this target do we actually have per microliter. And so, don't focus on the numbers here, but if you just look up here on the left-hand side, we obviously have a problem, right? What I said before was that in order for digital PCR to work, you have to start seeing negative wells, no copies show up in order to be confident in this. What's happening in this case and over here is that we're just oversaturated. We distributed the sample across those subarrays, but we just ended up with so many copies per well. There's not enough negatives. There's no way that we can really go in with absolute certainty and say, okay, this was 1,000 copies versus 5 copies, right? Using traditional endpoint digital PCR approach. So this is actually the range of where the system will work, because now you can go in and actually get an accurate count, and we determined, for these different dilutions that we had about 15,000 copies per microliter. And this was actually pretty good, because these were viral standards that we generate through our other side of the business. It's called Acrometrics. They generate standards of all sorts, and they're all part of Life Technologies as well. So we already had a good idea of what the copy count should have been in this. So we just did this as a proof of principle. So I described earlier about low-fold copy number determination. I think this illustrates here pretty well. If you're looking at trying to interrogate, like copy changes, let's say, like 4 to 5, 5 to 6, 6 copies versus 7 copies, the percentage change gets smaller and smaller and smaller. The reason why I'm showing this is because for those of you that are interested in studying copy number variation in genomes, we actually have a different line of TaqMan assays. They're called TaqMan copy number assays. So it's a completely different line of assays we developed that worked actually extremely well through traditional qPCR. You can use this on any real-time system. But the limitation, again, was the CT resolution. So our limit there was really 4 to 5 copy differentiation. We could determine 20-fold changes. The only way we could do that was running a high number of replicates. So we had to run technical replicates in order to get statistical confidence in the actual copy call. Completely impossible for us, regardless of how many replicates we used, when we went beyond 5 copies. So we tried over and over to determine 5 to 6 copy differentiation. Couldn't do that using our TaqMan copy number assays, but then digital PCR is an approach that you can use and to rescue that, to actually get accurate counts. This clicker is starting to die. The overall workflow is extremely simple. There's nothing novel, again, to this. I told you before, it's just as simple as just taking your sample, the master mix, whatever assay that for the locus you're trying to interrogate, how many copies you have. You can actually mix in a reference assay as well simultaneously. The system can actually detect on 2 targets. The reason why you would run a reference assay with the Vic fluorophore is that so you could be certain that when you actually look at the copy counts, that the copy counts that you got are referenced back to a control input. So let's say something like if you knew that RNase P was present in your sample at a certain number of copies, or if GAPDH, for example, was non-changing, if it's a static expressor, you can actually use that as a control. Because if you didn't use a control along with the target that you're trying to interrogate, you could easily get a false negative assumption, right? Because you could just look at the results and say, oh, I have no HIV copies present in the sample. Well, how do you know there was none there? It could be that you just didn't add anything to those wells, right? So you use a control to actually guarantee that, yes, we have sample introduced, because that's being exhibited by the copy counts of our control gene. And so that's the approach that we recommend, is actually using a two-target approach. So the machine can actually count with both. You just mix all this together. The system actually loads up the array for you again, pop it into the system, and this is the actual readout. And I want to focus on this now because I told you, I kind of lied a little bit. I said, digital PCR is just simple distribution, and it's just endpoint determination, right? You're just basically distributing, running PCR, and then just getting a count. I forgot to mention why we call it digital PCR. Did anybody figure out why it's called digital PCR? It's just a binary determination, right? Yes or no, yes or no. Is the well black or did it light up? So that's why we call it digital PCR, like ones and zeros with computers. Now, where we're a little bit different here with this digital suite analysis software, what actually sets us apart, digital PCR is a really hot area right now. There's a lot of our competitors are in this market. How we're differentiated from them is that we actually combined endpoint digital analysis with real-time data as well. So we enable you to actually capture the real-time CT plots and then go back and determine if the data that you're getting is actually real by combining a CT value. So by using that approach, what we can do is this. So we can use digital PCR together and couple it with genotyping, a genotyping run, or it's a gene expression run, right? We can go back and say, okay, not only do we get an endpoint analysis, we're trying to determine basically, what these, miscalls or like off calls are. Is this really a different allelic variant? Is there something else going on? Was our copy number variation present in the actual polymorphism in that locus? And is that what's actually thrown off these clusters? Or is this just a random, spurious amplification event that has absolutely nothing to do with what's really present in my sample? So by combining digital PCR with the genotyping cluster calling or gene expression data, we can go back and look at these two data points as an example and say, okay, was this real or was this not real? We go back here and say, did we see a CT value? Did we actually see an amplification plot come up? Did we see exponential rise? In this case, we didn't, right? It's actually pointing to this area here. It's hard to see, but area over here where there's no amplification. So whatever this fluorescence was just something that had nothing to do with true amplification of your target. So it allows the researcher to go back and actually rescue ambiguous results. Right? This was never really possible before. And then with this, you look at this one sample that actually occurs off cluster here. Well, you get confirmation by going back and looking at the actual amplification curve that was generated. So that's what actually sets us apart from the competition. A lot of the other digital PCR providers only provide you the endpoint analysis. They only provide you this view because they don't capture the data in real time as well. Whereas we allow you to go back and rescue ambiguous results. So that's the key point there. I need a new battery. I'll start concluding the talk here by talking about somatic mutation detection because I think this is a nice segue into our upcoming talks. That Andy will be delivering on the Ion Torrent and the AmpliSeq cancer panels. Tony actually mentioned those this morning as well. But somatic mutation detection is something of extreme interest, obviously. So we, from a next generation sequencing standpoint, but then also from a qPCR standpoint, recognize the need to come up with assays that can actually interrogate these rare variants. These somatic mutations, they're actually present, let's say, in a tumor sample at a very, very small frequency or small percentage. If this mutation is off by a single base, it's just a single base polymorphism, how can we actually interrogate and recognize that polymorphism when it's in the presence of a dominant wild-type background? A dominant wild-type background that's identical genetically with the exception of a single base. How can this assay actually differentiate and tell you that that's the true mutant signal? So we developed what we call Cas-PCR, or the mutation detection assays, to answer that question, as well as questions of let's say like circulating tumor cells. So there's a lot of interest now in just from a blood draw, looking at these few cells that are actually just wandering through the bloodstream. They could be as few as just like 1 or 2 cells, like floating around in a milliliter of blood. how can we actually get assays, these rare tumor cells, detected with enough sensitivity. So we embarked on specific TaqMan PCR. This is actually different from allele-specific PCR, which has been described in current protocols or the Maniatis books. Allele-specific PCR was used as much as like 20 years ago. But where Cas-PCR is actually different from allele-specific PCR is that we did a little twist on the assay itself. So in the assay, what we have is we have a probe, and I have it shown here as it says ASP. That's an allele-specific probe. So that's the probe trying to detect our actual mutant target. The probe actually terminates at the 3' end right above the mutation. So we expect that if it was not complementary, that the 3' end base would be floating off, right? It wouldn't be sitting down perfectly, and it wouldn't amplify. But we know in reality that oligonucleotides, with hybridizations, things are always like coming on and letting go, coming on, letting go. You will still get significant amplification, even if there's a mismatch of that base right there. And in that same assay, we added also a blocker. So where it says ASB, that's an allele-specific blocker. This is an oligonucleotide that's complementary and will sit down to your wild type region. So basically what happens is it effectively blocks or quenches that, the wild type genotype. So what that does is basically allows us to just PCR amplify only the mutation, even though the mutation is present at very, very, very low frequency. Because if we didn't block the wild type out, it would still amplify, right? This specific target, it still would sit down and give a significant amplification. So basically, that's how we came up with these Cas-PCR mutation detection assays. We're very, very efficient at picking out these rare, like a needle in a haystack, this rare variant. Example here, I mentioned allele-specific PCR. So if you just basically look at single base mismatches, obviously here we have a GC perfect match, right? So let's say that 3' base was a perfect match. We're getting a CT value of 27 cycles. If we had a mismatch, a deliberate mismatch of a GT, you can see that we're actually only delaying the amplification by a single cycle. We're still getting significant amplification. So if you compare that now to this Cas-PCR approach, where we use this blocker to block out the dominant wild type background, what happens is this. In the presence of the mismatch, we almost completely, we just kill the PCR entirely in the mismatch. We get the CT matches when it's perfect. So this is basically just, we've demonstrated this, and these assays were released actually about a year ago. We've actually built onto this list, because what's happening now is, the story again that we're trying to tell you is the entire genetic analysis workflow. Basically going from hypothesis-free discovery on the ion torrent systems, which you'll be hearing about this afternoon, and then moving towards more focused pinpoint validation using qPCR. So these are like two huge parts of our business that are without a doubt complementary. And so we're trying to match the content that we're seeing on the ion torrent side of the business with all their AmpliSeq panels. Right? So all the mutations that they're showing on those panels, we want to give researchers the ability to go back and look at those same mutations and validate those mutations using our Cas-PCR assays as well. So we're actually building to this list. Right now we have 285 mutations, but we're building and building. So we have a perfect match to the PGM AmpliSeq cancer panels. And you saw this slide earlier. This is actually a duplication of Tony's slide from earlier. But this is just highlighting what I just said a second ago, that we really want to present like the entire workflow for the researcher. The ability to just come in, use the PGM, and run these AmpliSeq panels, and basically just take those results very easily in a streamlined fashion, move from that system over to QuantStudio and do validation there seamlessly. And not only, of course, is this for the mutation assays, this is for basically any area of research, genotyping, microRNA profiling, looking at, let's say, non-coding RNAs. A lot of interest lately in understanding what non-coding RNAs do. All this non-coding RNA information is being generated, of course, from next-generation sequencing. And so we provide a way of actually using TaqMan assays that are specific for all these non-coding RNAs. We design them all actually already in advance. And basically, we provide those on our websites for researchers to validate and complete the entire workflow. And again, this is Tony's slide too, I stole this. So this is just to complete the whole message and tie in the story of what Tony started, what I talked to you about today, and then what our speakers this afternoon will be talking about as well when we talk about next generation sequencing. We really wanted to provide the entire solution for the researcher, the entire workflow.